In this video, I'll be talking about the luminescent bacterial in detecting cadmium in underground water. Just in case you're not a biochemical engineer, there might be some jargons in the presentation, and definition of them is here. So the problem is identified in Pakistan and Eastern Iran, where unsafe amount of cadmium is detected in the underground water. So first, how is this bacteria being able to survive under heavy cadmium conditions? This is done through the recombinant bacteria, where the lac operator is replaced by a cadmium-responsive gene. This gives the recombinant bacteria the ability to respond to cadmium. It also allows it to emit photons of light. So here is a drawing of the gene added to the bacteria. The first, the CAD promoter and CADC, is used for its resistance. And the second part, the LUCFF gene, is responsible for a reaction called the luciferin-luciferase reaction. In this enzymatic reaction, the luciferase is the catalyst and luciferin is required as a substrate. So the luciferin binds to the active site and ATP and oxygen is required to oxidize luciferase into inactive oxyluciferin. And during this process, photons of light is also created. So as mentioned, there are photons of light created, and these are used to convert the response of the recombinant bacteria into quantitative data, where a luminometer measures amount of photon and it's detected by the PMT. And this converts photons into electrons and it's measured in relative light units. This means that where there is a higher relative light unit, there's more response to cadmium, hence there is a higher concentration of cadmium detected. There are also pros and cons to this technique. Firstly, the use of bacteria allows a easier genetic modification. It also has a higher mobility, meaning the bacteria can be genetically modified in other countries or cities with qualified laboratories, then transported to rural cities such as Pakistan for a higher accuracy or preciseness. It's also more cost and time saving, but it also has limitations. For instance, the least cadmium concentration found in Pakistan is as little as 0.417 macromole per liter, whereas the recombinant bacteria can only respond starting from 1 macromole per liter. This can be solved by adding a known amount of cadmium so that the concentration raises, and then when the data is collected, the known amount of cadmium can then be subtracted to find the actual amount. The second limitation is that it has a reduced accuracy, whereas zinc ions and cadmium ions has really similar property and sizes. This can be solved simply by filtering out the zinc ions in advance so that the cells will not get mistaken.